Here in chapter 4, we're mainly going to focus on bacteria. Of course, it also mentions archaea, but again, since we're talking more about medical micro, we're mainly focusing on bacteria and a lot of their characteristics. So first of all, again, just a recap, the difference between prokaryotic and eukaryotic cells. So bacteria and archaea are both prokaryotic, which means that the packaging of their DNA, so their cells, so both prokaryotic and eukaryotic cells have DNA, but in bacteria and archaea, again, they do not have that nucleus. There's no organelle, no membrane-bound compartment that stores the DNA. It's just hanging out inside of the cell, whereas eukaryotic cells have that organelle to store their DNA, so they have a nucleus. In addition, there are other internal structures that are different. Again, the bacteria and prokaryotic cells don't have those complex membrane-bound organelles, whereas the eukaryotic cells do, in addition to the nucleus, have other organelles like a mitochondria, the Golgi, the endoplasmic reticulum, many other types of organelles. So just kind of looking at, again, the basic cell structure for bacterial cells. So all cells, regardless of whether they're prokaryotic or eukaryotic, have a cytoplasmic or cell membrane. So that's a phospholipid bilayer that controls what's coming in and out of the cell. So again, all cells have that. All cells also have ribosomes, which is where proteins are made. They have a gel-like fluid inside called the cytoplasm. And then again, they have some type of chromosome in it, or DNA. So again, all cells have a cell membrane, they have a cytoplasm, ribosomes, and DNA. So some type of chromosome. So in a typical bacterial cell, inside, that's again where you're going to find that cytoplasm, the ribosomes to make those proteins. Again, those are not organelles, they're just structures. Inclusions, and those are typically used for kind of like storage. Again, the chromosome or nucleoid, that's where the DNA is going to be found. They have some type of cytoskeleton, so some type of framework, but it's much less complex than in a eukaryotic cell. Some of them can form endospores, but not all bacteria can. Only a couple different types can form this really dormant state called an endospore. And then a lot of bacteria also have what are called plasmids, and these are extra pieces of DNA that provide some type of advantage. So it's not required for the cell to exist, but they're extra pieces of DNA. They're outside of the main chromosome, and they're called plasmids. And then some of them can even have tiny micro compartments, but again, not to the complexity of an organelle. And so that's everything contained inside of that cytoplasmic membrane. And then most bacterial cells outside of the cell membrane have a rigid cell wall, because these are mainly single-celled organisms. So that cell wall provides some protection for the cell. So again, it's really rigid. It keeps them from just popping or breaking. So again, some kind of a protective layer on the outside called the cell wall. Again, not all, but the majority of bacterial cells will have that cell wall. And then again, on the outside, so the external part, it can have various things. And again, these are optional. Not all bacteria are going to have all of these. They may have one or two. They may have them all. They may have none of them. But sometimes they have a surface layer, kind of like a slime or an S layer, or what's called a glycoclax. It's kind of like a sugary layer that helps them stick to things or like to move or glide past things. And then they can also have different appendages. So for example, a flagella, which is what we see here. It's a long tail-like structure, helps it to move. They can also have some shorter projections called pili that we see here that provide various functions. Again, some finger-like projections called fimbriae, and some of them even have these nanowires or nanotubes. So those are things outside of the cell that provide some type of advantage. So here we're looking at things in a little bit more detail, and I know this is pretty small, but in the PowerPoint that you can download, you can always expand it or blow it up, zoom in. It's also in your ebook. But again, things that all cells, this is all bacteria, but this is actually all cells, prokaryotic or eukaryotic, things that they have. Again, that cell membrane that we talked about, again, that's the outer layer, determines what's inside and outside of the cell, controls what's coming in and out. They have that chromosome, the DNA that's in them. Bacteria only have one big chromosome. And so that's contained there. Our cells have 46 chromosomes. So we all have DNA, just different amounts. And then the ribosomes, again, that's where proteins are going to be assembled. Again, all cells need to make proteins. They are a little smaller in prokaryotic cells, so they're a nice target for antibiotics. And then again, they also have the cytoplasm, that kind of fluid inside that allows kind of a storage for enzymes, but also just kind of like a gel-like fluid inside for stability. And then again, some bacteria, some of these we already mentioned, again, that S layer, kind of like a slime layer, that can be used for protection or for attachment, again, kind of like a sugary layer. Fimbriae and pili, kind of the same. So these are kind of like bristles that extend out that just helps them to attach to different surfaces. Some of them, so we'll talk about gram-positive and gram-negative cells. So gram-negative cells also, in addition to the cell membrane, have another membrane outside of that called an outer membrane that has various structures in it. Again, most bacterial cells have a cell wall, a rigid layer made up of a compound called peptidoglycan that gives them a nice little protection. Again, some type of cytoskeleton inside that provides a framework for it. They can have various pili, so single is a pilus. Those can be used for attachment, but they can also transfer DNA. Some of them have a capsule, again, just a slightly different structure than the S layer that provides some attachment, kind of a sugary layer that can provide some protection as well as adhesion. Again, those inclusions are granules that's used to store nutrients for this cell. So whenever they don't have as much, they still have kind of a backup. Some of them can have those micro compartments, little kind of protein coated packets there that collect different enzymes that are there. Again, they're not organelles, but they're kind of smaller compartments. Again, a lot of them have plasmids, those extra pieces of DNA. They're circular. Again, the idea though is they have extra genes. These are not genes they have to have. They're extra things. And then some can also have a flagella, more of a long tail-like structure that helps them to move. This one shows the bacteria only having one, but some of them have more than one flagella on them, but that helps them kind of like a whip, whips back and forth and helps them move. And then a couple others that aren't shown here. So again, that endospore, that dormant state, again, only some bacteria can produce that. Some have some membranes that go inside of the cell kind of more randomly than we see in eukaryotes. And then again, those nanowires or nanotubes, which are kind of like membrane extensions that help the bacteria transmit electrons or nutrients to other bacteria. So kind of like an extension to where they connect to another cell and can kind of transfer nutrients or even electrons. Right, if we look at some shapes of bacteria, there are three main shapes. Coccus, which are kind of circles or ovals, bacillus, which are going to be more kind of club-shaped or rod-shaped, and then also curved, so especially Spirillum or Vibrio, kind of like a comma, corkscrews. So essentially round, kind of rod-like, or curved are the main shapes. And some are actually pleomorphism or morphic, which just means they can have a variation in size and shape. So it's a bacterial cell that's not always coccus or it's not always bacillus. It can be multiple different times. So they kind of change shape based on genetic differences or different nutrition that they have. So this is just showing, again, the coccus here, kind of the rod-shaped. Again, our rods or bacillus. And then some of the more curved and some of the variations. And a lot of times this is used in the naming, so like Staphylococcus, that tells us that these are going to be circles. Streptococcus, we'll come back to these, so that also indicates something about their arrangement. So Staphylococcus are kind of clusters. Strep indicates chains, so there can be Streptococcus or Streptobacilli, Staphylococcus, Staphlobacilli. Again, so a lot of times the name of the organism gives an idea about the shape that's in there. So again, our Streptobacillus These are long chains of those rod shapes. Or the vibrio, again, this is kind of that comma shape. We can have some corkscrew, kind of spirillum that fall in that curved category. So again, just some of the common arrangements that we see. So there are diplococci, so diplo meaning two. So these are pairs of circles. Tetrads would be a group of four. Staphylococci are clusters, so again, kind of like grapes. Strepta, those are chains of cells. And then Sarcinia, which are more cubed shaped. And again, a lot of times we'll see this in the name, but also it can be used in characteristics when you're looking at various organisms. And then some of the other ones, so again, in the bacilli, they're diplobacilli, in pairs, streptobacilli, chains. There can be palisades that occur where they kind of fold back and forth, so they have side-by-side rows. So they're kind of like stacked on top of each other. And then spirilia sometimes can be seen here in short chain, and then spirochetes rarely remain, though, attached after they divide. All right, now a really cool, but unfortunately usually not a good thing in the medical realm or healthcare, a collection of kind of a community of microorganisms is referred to as a biofilm. And these are bacteria that often live cooperatively with other organisms. And sometimes they can be the same species, so just lots of the same, but a lot of time it's other organisms and not even just bacteria, but it can be archaea, fungi, algae. So the picture that we see over here, this is a variety of different microorganisms that are living in this community that make up what we call a biofilm. So they're essentially a microbial habitat. They're typically pretty structured, so they have access to food. There's usually kind of tunnels that go through them. They'll let food and water and different gases in and out, as well as waste to be removed. So again, they provide benefits for all of them. So it provides protection. Again, they can kind of work together in this microbial community. But unfortunately, in the medical field, they're also really hard to get rid of. They're hard to treat off different surfaces that are trying to become sterile. Antibiotics have a hard time reaching all of them because they can kind of hide within the biofilm. So again, this community is referred to as a biofilm. So how they form, so they form typically on moist surfaces. So we think about whenever you wake up in the morning, all that gunk on your teeth, that's a biofilm. So kind of the moist habitat of your mouth. So they start on those, steady surfaces that have a thin layer that's slightly sticky material. So that way, if you eat a lot of sugary foods, you're more likely to have more of that buildup and that plaque because it feeds them. It allows them to stick. So it allows that first single cell to stick, and then it starts to multiply. So we see that here. So here's our surface. We have some kind of free-floating bacteria here, but then they're allowed to stick. So we get one or two that stick to the surface, and then they start to multiply. So then they start secreting that sugary layer, like a capsule or an S layer or something outside of their cell, and that allows all these other organisms then to start sticking. So they secrete that substance, and that attracts other ones to start coming in. They secrete material that allows others to come in. So they end up having this variety of thickness. They are often stratified, meaning they're kind of in layers that go out there. But again, it just lets them build that. But again, here's that sugary layer called a glycoclax. Again, capsule, slime layers, all of these terms, just the sugary kind of substance that gets secreted so they can attach and attract all these other organisms. Now, some of the appendages, again, some of these we already, or all of these we already mentioned. Again, these are things sticking out of the cell. So the flagella provides that motility, allows it to move. The fimbriae, the pili, and the nanowires are somewhat similar. So there's shorter projections that come out, but they can also provide attachment or even channels to let things in and out. So here's an example of the flagella. Again, it's just a long kind of tail or whip-like structure that sticks out and allows it to move. And cells can either have a single flagella that we can see here in this, or they can actually have lots of them where they're coming out all over. Now, some of the appendages on this section enhance their pathogenicity. So again, pathogen is a microbe that can cause a disease. Pathogenicity is how well it is able to cause disease. So pili, or pilus is singular, pili plural, it allows adhesion. It doesn't let it move like the flagella, but it allows it to attach. So one of the first things the microbe has to do to cause an infection is to attach to a surface. So the pili can help with that. Fimbriae is another appendage that can occur that does the same thing. They're just a little different structure. Flagella can also be used a little bit for attachment in some. It's mainly we think about movement, but they can be used for attachment as well. And the nanowires or nanotubes, again, are other things that can allow microbes to attach to each other, create that channel, helps with the biofilm formation, but also exchanging nutrients that allow them to cause disease. Now, just a little bit more about the fimbriae. These are small bristle-like fibers that sprout off. Again, what they're made up of varies, but most of them are made up of a protein, and they have a tendency to kind of stick to each other and to surfaces. So not only do they help them attach to a surface, but they kind of stick together, stick to other microbes, and again, can be helpful for them in making those biofilms. So for example, E. coli, Escherichia coli, uses those fimbriae to attach to epithelial cells that allow them to cause disease. Now the pili are a little bit longer than the fimbriae. They have a tube-like structure, also made up of a protein called pilin, so pili, pilin. But these are only found on gram-negative bacteria. So they're not, again, what we've been talking about are not on all organisms, only some of them, but these pili are particularly only found on gram-negative. And they can be used in what's called conjunction. We'll come back to this a little later in the semester, but that's what this is showing here, this conjunction. So the pili here that are attaching to microbes, another one here. So this pili, what happens is that some of the plasmids that are in here, they can actually move through that pili and over to another cell. So that allows them to transfer DNA, and it's particularly an issue with antibiotic resistance. So if one bacteria is resistant, it can share that gene with another through this transfer through the pili. So that increases that pathogenicity, its ability to cause disease. And again, other pili can be used for attachment to help them attach to surfaces. Now the coatings, we talked about the S layer and the glycoclax. There are a few others as well. So these are some coatings, again, similar to the appendages, they're just slightly different. The S layer here, this is a bunch of proteins that are linked together, provides protection from environmental conditions. So if it's like really fluid, there's a lot of water, the salt concentration changes. So these only get made whenever there's a hostile environment. So for those that can produce an S layer, it's only whenever it's needed. It's not there all the time. Others can make what's called a glycoclax. So this is a sugar unit, so a polysaccharide or like glucose is an example that may or may not have protein as well. Again, it helps it with attachment. A slime layer, this forms kind of more loosely around the cell, protects it from losing water or nutrients. So it's more a protective layer like the S layer, not so much with like pathogenicity and causing disease, but just an advantage to help it in different environments. One though that we will talk a lot about with pathogenicity is the capsule. Unlike the slime layer, it's more tightly bound to the cell. It's denser and thicker, made up of those polysaccharides, but that is definitely one that kind of protects and hides the cell from our immune system that is looking for bacteria, as well as helps them attach to increase their ability to cause disease. And this is just kind of showing the difference between like a slime layer, here again a little loosely packed, not real tightly associated, versus that capsule where it's real dense, very compact structure that's there. And this is just showing under the microscope, but it looks like, again, this is the cell body, and this is how thick that capsule is. So it's much thicker than the cell itself, but so it helps protect it. So if there's an immune cell out here looking for things, that bacteria is hidden underneath that capsule, so it makes it easier for it to hide from our immune system. So again, just some of the specialized functions here of our glycoplaques, particularly in the capsule. Again, as we mentioned before, a lot of pathogenic bacteria, and those are the ones that can cause disease, have a capsule because it not only helps them stick, but it protects them against phagocytic white blood cells, which are part of our immune system. We'll cover later in the semester. But again, they are the ones that engulf bacteria trying to break them down. But if they have this capsule, it makes it harder for them to get engulfed. And then also that glycoplaques helps to form those biofilms that are beneficial for them, causing disease as well. So for example, the plaque on our teeth that we mentioned before, that sugary layer on the outside helps protect them. And it also, as far as sterilizing, can infect long-term indwellings of artificial devices, especially like respirators, catheters. They can become infected with these biofilms that make them very hard to sterilize and allows them to be able to cause disease.